Sir, live paging, sir. So I'm sharing the link in the chat. Live, sir.
ஆனா ரொம்ப இதே இல்லை, ரொம்ப வால்யூம் கூட்டினா தான் வருது. அதனுடைய இதே இல்லை.
அந்த இதை கூட
சென்சிட்டிவ்
மியூட் பண்ணிடுங்க எல்லாத்தையும் பேசும்போது. ஸோ ஒருத்தரா மீட் பண்ணிட்டு இருக்கிறது.
அந்த லிங்க் வேற யாருக்காச்சும் குரூப்ல போடணும்னா எடுத்து போட்டுடலாம். நம்ம லிங்க் இஸ் ஹியர்.
ஆரம்பிச்சலாமா?
ஆரம்பிச்சலாங்க. அனைவருக்கும் வணக்கம். நம்ம இங்க எல்லா உலகத்துல இருக்க எல்லா பகுதியில இருந்தும் ஆட்கள் வந்து இந்த ப்ரோக்ராமுக்கு வந்திருக்காங்க. அதை பார்க்கும் போதே ரொம்ப சந்தோஷமா இருக்கு. இந்த நிகழ்ச்சி வந்து ரொம்ப நாளைக்கு முன்னாடியே வந்து அரசையா வந்து பிளான் பண்ணிட்டு இருந்தாரு. இன்னைக்கு வந்து அது வெற்றிகரமாக நடைபெறுகிறது. அதுல ஒரு சந்தோஷம். இதோட பின்னோட்டம்னு பார்த்தோம்னா வந்து இப்ப 20 செப்டம்பர் 1924 அது ரொம்ப முக்கியமான நாள். அன்றுதான் வந்து சார் ஜான் மார்ஷல் வந்து இண்டஸ் வேலி சிவிலைசேஷன், சிந்துவெளி நாகரிகத்தை வந்து உலக வந்து தெரியப்படுத்தினாரு. அன்னைக்குல இருந்து பார்த்தோம்னா நிறைய வந்து அதை தொடர்ந்து அகலாய்வுகள் நடந்துட்டு வருது. அதே மாதிரி வந்து கிடைச்ச பொருள்களை வச்சு நிறைய வந்து அறிவியல் சார்ந்த முடிவுகள் நிறைய வந்து வெளிவந்திருக்கு. இதை தொடர்ந்து பார்த்தோம்னா இந்தியாவோட வரலாறு வந்து வேறு விதமா வந்து கட்டமைக்கப்பட்டு கொண்டே வருகிறது. இப்ப அதே அந்த அந்த அறிவிச்ச நாள்ல இருந்து பாத்தீங்கன்னா இன்னைக்கு வந்து 100 ஆண்டுகள் ஆகுது. இந்த 100 ஆண்டுகள் ஆனதை வந்து நம்ம வந்து வெகு சிறப்பா சிந்துவெளி நூற்றாண்டு கொண்டாட்டங்கள்னு பலவேறு தளங்கள்ல வந்து நடத்திக்கிட்டு இருக்காங்க. அதே மாதிரி இந்த ஜேர்னி ஆஃப் அ சிவிலைசேஷன் ஸ்டடி சர்க்கிள்லையும் நாங்களும் கொண்டாடணும்னு நினைச்சோம். அதோட ஒரு நிகழ்ச்சியா தான் இன்றைய நிகழ்ச்சியா வந்து அமைஞ்சிருக்கு. ராஜையா வந்து இது ஜெனட்டிக் ஹெரிட்டேஜ் ஆஃப் கரண்ட் டே தமிழ்ஸ் என்ற தலைப்புல வந்து நமக்கு பேச இருக்காங்க. அவங்கள வந்து அவங்களோட வர கட்டாயமா வந்து ரொம்ப நல்லா இருக்கும்ன்றது கூட இருக்க எல்லாருக்குமே நமக்கு தெரியும். ரொம்ப வந்து ஆழமான உரையா இருக்கும், தகவல் நிறைஞ்ச உரையா தான் இருக்கும். அதையெல்லாம் நம்ம வந்து கேட்க தயாரா இருக்கோம். நம்ம எல்லாரையும் வந்து ஒரு வரவேற்கிறதுக்கு அரசையாவை கூப்பிடுறேன். அரசையா வந்து அவரும் வந்து சாதாரணப்பட்டவர் இல்லை. அவர் வந்து ப்ரொஃபஸர் ஆஃப் பயாலஜி அட் யூனிவர்சிட்டி ஆஃப் மேரிலாண்ட். அது போக வந்து அமெரிக்க தமிழ் அகாடமி அப்படின்ற ஒரு வந்து ரொம்ப நாட்களா வந்து நடத்திட்டு வராரு. கிட்டத்தட்ட 10,000 குழந்தைங்களுக்கு வந்து தமிழ் சொல்லி கொடுத்துருக்காரு. அதாவது அமெரிக்கால பிறந்த குழந்தைங்களுக்கு வந்து தமிழ் சொல்லி கொடுத்துருக்காரு. அவங்க பேச, எழுத, வாசிக்கன்னு வந்து நிறைய முயற்சிகள் எடுத்துக்கிட்டு இருக்காரு. அது தமிழுக்கு ஒரு பெரிய தொண்டுன்னு தான் நான் நினைக்கிறேன். அது போக அரசையா வந்து ஒரு சீரிய பெரியார் பற்றாளர். அதுலயும் வந்து அவரோட பகுத்தறிவு சிந்தனைகளையும் அப்பப்போ வந்து எல்லாரோடையும் பகிர்ந்துக்குவாரு. ஐயா, நீங்க எல்லாரையும் வர வைக்குவாங்க.
வணக்கம், எல்லாருக்கும் வணக்கம். உங்களை இந்த சிறப்புமிக்க ஒரு உரை நிகழ்ச்சிக்கு வரவேற்கிறதுல ரொம்ப மகிழ்ச்சியா இருக்கு. இப்போ ஒரு சிறு வரலாறு இந்த நிகழ்ச்சியை பற்றி முனைவர் பாமா சொன்னாங்க. எல்லாமே முனைவர் ஆர் பாலகிருஷ்ணன் ஐஏஎஸ் அவருடைய கடும் உழைப்பு, 30 ஆண்டு கால உழைப்பு, தரவுகளோட நிறைந்த தரவுகளோட வெளியிட்ட இண்டஸ் டு வைகை, ஜேர்னி ஆஃப் அ சிவிலைசேஷன் இண்டஸ் டு வைகை அப்படிங்கிற ஆங்கில புத்தகம் வெளியிடுவாங்க போது அதை நம்ம தமிழ் மாணவர்கள்லாம் படிக்கக்கூடிய மாணவர்கள்லாம் படிக்கணும்னு சொல்லி ஒரு பெரிய புத்தகத்தை பல பாகமா பிரிச்சு படிச்சு முடிச்ச பிள்ளைகளுக்கு சான்றிதழ் வழங்கணும். அதுல அமெரிக்கன் தமிழ் அகாடமி, அப்புறம் வட அமெரிக்க தமிழ்ச்சங்க பேரவை, ஃபெட்னா ரெண்டும் சேர்ந்து அந்த நிகழ்ச்சியில நடத்தினது. அதற்கு பிறகு பெரியவர்களுக்கும் அவர்களுக்கு நடத்தினது. இதுல இந்தியா, அமெரிக்கா, கனடா, சிங்கப்பூர் இந்த மாதிரி பல நாடுகள்ல இருந்து எல்லாரும் கலந்துக்கிட்டாங்க. இதுல சிறப்பு என்னன்னா, அது முடிந்த பிறகு இங்க உள்ள சில புத்தக குழுக்களும் படிச்சிருந்தது. வேள்பாரி, ஏர்லி இந்தியன்ஸ் இந்த மாதிரி புத்தகத்தை படிச்சவங்க எல்லாம் இப்ப நிறுத்திட வேண்டாம், இதை ஏதாவது தொடரணும் அப்படின்னு அது பேரே ஜேர்னி ஆஃப் JOC 24. அவங்க முனைவர் பாமா முத்துராமலிங்கம் அவர்கள் சொன்ன மாதிரி இதை ஒரு 100வது ஆண்டுங்கிறதுனால இன்னும் சிறப்பா இதை ஒட்டிய தலைப்புகளில் பேசுவோம். இப்ப எங்க அமெரிக்காவில் உள்ள அடுத்த தலைமுறைக்கு தமிழுடைய வரலாறு தொடர்கிறது, அதை பற்றி தெரியப்படுத்தணும்னு சொல்லும்போது பெரியவர்களும் இதை நாம தொடர்ந்து வாசிக்கணும், தொடர்ந்து இதை பற்றி பேசணும், கருத்து பரிமாறணும்னு சொன்னார்கள். இதற்கு முந்தைய உரை நிகழ்த்தினது நம்ம முனைவர் பாமா முத்துராமலிங்கம், இப்ப இணைப்புரை வழங்கிட்டு இருக்கிற முனைவர் பாமா முத்துராமலிங்கம். அவர் IIT சென்னையில PhD படிச்சுட்டு எலக்ட்ரானிக்ஸ் அண்ட் கம்யூனிகேஷன் துறையில குவால்காம் அப்படிங்கிற நிறுவனத்துல சிறப்பா பணியாற்றிட்டு இருக்காங்க. அவங்கதான் போன உரை நிறுத்தினாங்க. இதுல போன உரையில அவங்க லோத்தல் தொலவீரா போய் பார்த்துட்டு அந்த உரை நிறுத்தினாங்க. நம்ம ராஜ் முத்தரசனே முன்னாடி என்ன பேசிட்டு இருந்தாருன்னா, நம்ம இந்த இப்ப பேசிட்டு இருக்கிறோம், நம்ம எல்லாரும் ஒரு பயணம் மேற்கொள்ளணும் அப்படின்னு சொல்லிட்டு இருந்தோம். அவங்க நேரா பார்த்துட்டே வந்துட்டாங்க. அந்த வகையில ரொம்ப சிறப்பு. அவங்க இப்போ இணைப்பை வழங்குறதும் ரொம்ப சிறப்பு. ராஜ் முத்தரசனை பத்தி அவங்க முறையாக அறிமுகப்படுத்த இருக்கிறாங்க அவங்க பேசுறதுக்கு முன்னாடி. அதுக்கு முன்னாடி நான் ஒண்ணு சொல்லணும். பேராசிரியர் ராஜ் முத்தரசன் அவர்கள் பெட்னா அப்படிங்கிற அமைப்பு, தமிழ்ச்சங்கங்களின் கூட்டமைப்பு, அமெரிக்காவில் நிறைய பேருக்கு தெரிஞ்சிருக்கும். அந்த கூட்டமைப்பு 1988 அவர் ஃபவுண்டர் பிரசிடென்ட். அதனால அதுல இருந்து தொடர்ந்து தமிழ் சார்ந்த ஆலோசனைகள் வழங்கிட்டு இருக்காரு, தமிழ் சார்ந்து இயங்கிட்டு இருக்காரு. இங்க இந்த அமைப்புல இப்ப இன்றைக்கு குடியுரிமை உள்ளவர்கள் எல்லாருமே ஏதாவது ஒரு வகையில தமிழுக்கு தொண்டு செய்து கொண்டிருக்கிறார்கள், அமைப்பு சார்ந்தும் அமைப்பு சாராமலும். With these few words, I really welcome all of you for today's a very interesting topic which we have been communicating through WhatsApp for this wonderful lecture. Welcome you all.
Thank you, Aya. Before we get into the formal talk, let me introduce Professor Raj Mutharasan formally. Professor Raj Mutharasan received his bachelor's from IIT Madras, India, and his PhD from Drexel University in 1973 in the field of chemical engineering. He has directed over 30 PhD dissertations and over 40 MS theses. His research has focused on process control, mathematical modeling of chemical engineering systems, biochemical engineering, process metallurgy, and recently on biosensors. He has been funded by such agencies such as NSF, NIH, EPA, USDA, DHS, and private companies. He has been awarded fellow status by ASHE, AAS, and AIMBE. He has served on the editorial board of Metallurgical Transactions, Biosensors, Letters, Applied Biochemistry, and Biotechnology. He has received such awards such as Drexel's Research Award, 106 Award, Distinguished Service Award, and Lindback Teaching Awards. He has published over 150 peer-reviewed publications and has been awarded some 12 patents, bulk of them being on his invention on cantilever biosensor for cancer research and pathogen detection. He served as an interim dean of College of Engineering, as a program director at NSF, and was awarded Frank A. Felcher Professorship in 1999. He received Distinguished Alumni Award from IIT Madras in the year of 2014 and has served on the Scientific Advisory Board of ALCOA, MMT Division, MPT Technology, Biochemical Technology, Cultural Division Technology, and so on. So this is his career profile, but he does more than this. As Professor Arasu Chelea mentioned, he has been the founding president of FETNA. Along with that, the genetic studies have been spearheaded by Spencer Wells in the late 90s. And his interest in genetics started with that. He goes back in the late 1980s, and in history and archaeology, he has an interest spanning over 25 years. More importantly, he has closely worked with Professor Spencer as well as Professor Pichappan of Madurai Kamaraj University in the DNA studies of the Tamil population. This says how eminent our speaker is. So we look forward for your enlightening talk, sir. Over to you.
மியூட்ல இருக்கீங்க.
Oh, I was muted. Okay. Thanks, Bama, and thanks, Arasu. Apparently, I was muted before that, so what I had said earlier did not reach all of you. So after hearing what Bama said, what I said was I should be turning up my mustache further up rather than down, right? Anyway, it's been an exciting area, and of course, as a teaching person, I've always learned that when you teach, you learn. That is, teaching and learning are pretty much occurs simultaneously. So I've learned a lot of stuff because over the last, although I have read in this area for a long time, but being able to put a cohesive talk that way to reach people requires for me to integrate it and then figure out how to say it to the people that way it'll reach the people that the audience well. So that made me work on this. So Arasu Chelea made me work till midnight and past midnight for the last few days. I put my final slides together last night about 12 o'clock before I went to bed. So this title was Genetic Heritage of Current Day Tamils has a context to Indus Valley civilization. And because there's a growing discussion about this topic, not only among Indian scholars, but also among the Western scholars as well. But of course, when I say India, it is only in the southern part of India. It is not omnipresent in every parts of India, unfortunately. But we'll see where that takes us. So the bottom of this, the graphics for this, by the way, Bama helped design it. Thanks, Bama. The bottom of it gives you the faces of Indus Valley civilization. That is, this is what we have obtained. Few more, but I've captured some of them. So an Indus Valley civilization, folks, don't look anything like any one of us look like in India at the moment. There are some exceptions. So that is to be kept at the back of our mind. And of course, all of this became very strongly evidenced in the work that was published just a few years ago based on Rakigargi excavation. Let me get into the talk. So my first disclaimer is I'm not an expert in genetics, archaeology, anthropology, or linguistics, but I've read extensively in this area. Right through my career, I have learned my topic, the area in which I do research, by self-learning, and that is part of a university professor's job, if you will. That is required for you to learn new things because the body of knowledge that exists is always expanding, and you have to learn new things in order for you to remain competitive in the research field, at least in the US. In the US, a faculty member is primarily known for his research, not for teaching, unfortunately, because research means in the university context, bringing in grants and money talks, as all of us know. So That has been the genre that I belong to. And I have dabbled in recombinant genetics with hybridomas about 30 years ago. That is because I was trying to improve yield of hybridomas that produce monoclonal antibodies. At that time, my lab was one of the pioneering labs in the US that dealt with cell culture cultivation that we can scale up to tens and hundreds of liters. We were written up in New York Times, for example. And that gave me that particular exposure gave me a foundational understanding of the topic that we're going to discuss today. Although I'm not an expert in genetics, I can appreciate the major things that are happening in that area. So what I shared today is sort of things that I've read over the last 20 years, which are very large volume of material. But I'm trying to condense all of that into a short 1-hour talk. And my journey goes back to 1990s because I participated in National Geographic magazine genographic project that was headed by Stephen Wells, and Professor Pichappan from MKU was participant in that project for the Indian section. And I should say, as I'm at this point, I should say the genetics of IVC folks and ancients of Tamil Nadu are yet to be determined. We have only one genome from IVC that was done just a few years ago. Nothing from Tamil Nadu, although we have rich collection of ancient skeletal remains. And I remember having a conversation with Professor Pichappan and Stephen Wells at the Tamil conference that was held at Chicago a few years ago. They have over 90 skeletons of antiquity that they would like to analyze, but there are bottlenecks that they have to overcome. Apparently, they have not been successful at this moment. So this gap, I hope some of you who listen to this talk will take this message forward and try to move the energies required so that we can get many of these analyses done, because I think we have rich historical data in the residues that currently exist in the Chennai Museum and elsewhere in Tamil Nadu. Anyway, with that sort of a broad disclaimer, this is my outline. So I will introduce you to very elementary heritage biology. There are nuances, but the major story is what I want to share with you. And then give you a historical perspective of archaeogenetics, because it has strong input into how history or the migration of people occurred and how it impacted the history of humankind, specifically of the Tamils, which is where my focus is. And then we'll pause briefly for questions and answers. And then we'll move on to the research report that was published about Indus Valley civilization that came out a few years ago, that opened up a lot of interesting new ideas. And of course, it said certain things that people have been talking about with very substantial evidence. So I'll share that with you. I'll read the key points of that article with you, share with you, and glad to answer questions. And then get into mapping of haplogroup L. This is one particular genetic marker which has been identified by many scholars as IVC folks. I don't think it was only a single L, single genome, but there are multiple, but this is one of the important ones. There was a very important paper that came out just a few months ago, which really mapped the migration of haplogroup L people. And that has important implications in the context of the work that was done with the recent Rakigargi excavation. So I'd like to call us those two body of knowledge so that to raise new questions and think about questions that we need to answer going forward. And then we'll pause for briefly and then get into haplogroup distribution in Tamil Nadu specifically. That'll tell you where the folks currently live in Tamil Nadu are from and what their genetic heritage is, which is the title of the talk. So the main topic is really shifted down to the fifth space. We'll stop there and discuss what I share with you then. And then I'll share my own DNA journey, because I've done a lot of my own work and I've consulted many ancient archaeogenetic databases and try to map my chromosome with what is available, etc. So there's a little bit of an interesting story to be shared. And I think that'll give you some idea as to how some of the hypothesis with the IVC and it supports Balakrishnan Ayya's hypothesis of Indus to Vaigai, but I like to call it Indus to Pennar because that's where my ancestors come from, near Kadalur area. And then finally, I'll open it up for, there are other heritage markers that researchers are looking at. I'll just give a very brief introduction to it that's still in research labs and they haven't come to the mainstream yet. And also try to remark a few things about future research needs in this area from the Indian context, because it's really wide. There are two dimensions I like for that to be. The toponyms work that Balakrishnan Iyer has done is wonderful, but we still need to do linguistics analysis, which is very, we have hardly scratched that area. And the other part is the ancient genetics of folks in India has not been done. All we have is one genome from entire peninsular India, and Indians occupy 25% of the world, and we have only one representative, and that should be overcome. Let me get into the topic. Okay, this is a very important chart published by Nature just last year, which is really a chart of the number of ancient genomes that have become available. Ancient genome meaning pre-Bronze Age, that is pre-3000 BC, 4000 BCE, and all the way up to 45, 50,000 years ago. And of course, this has been a major effort that has been going on in the US, for example, David Reich's lab, which is at Harvard, and also in Copenhagen. You know, Copenhagen was responsible for bringing together the some of the ancient hominins genome to the open space. And that's available, for example, if any one of you are interested, at the link that I've provided. I try to include links to every place where I have drawn my data from. That way, if you want to read further, you can go back to it. And I'll make sure the slides are available to people who requested via JOVC WhatsApp group or through RMRL through Sundar's group. And that way, you will always be able to go dig into what I share. That way, you can either verify or concur with the opinions that I might share. So bulk of the work has been done in Europe, Germany, and in Sweden, pretty much. There's been no work in this space that's been done in India, pretty much, in terms of laboratory work. And dealing with ancient genome requires a little bit of a skill, but it's a very easily learned skill. It's not too complicated. I'm sure David Reich will be very happy to share that body of knowledge with anybody who requests that. Spend 3 months in his lab and you'll be able to acquire that body of knowledge. And bulk of the sequencing technology is now automated with about $50,000 to $100,000 investment. You'll be able to do that in no time at all. On the other hand, when I had to do that sequencing 15 years ago, I'll deposit it in my central laboratory and I'll get my results after a week or so. And prior to that, in the 90s, I would send it somewhere else and I'll get my results after about a week or 2 weeks. So now this is almost like having a laptop instead of a mainframe computer to a cell phone computer, right? That the technology has become very readily available to an average user. Now, the discovery of the onset of the availability of ancient genome beginning 2015, it actually has an earlier history. The acceleration in this space is primarily due to new technologies that have come to the fore. And that has provided very important and significant insight into history of human migration and relationship between different population groups and etc. And within this, we have most of the information is about Europeans and Russians, and only about 500 about South Asian, South and Central Asians, out of the 500, only one in the entire country of India, which is 1/4 of the world's population, right? And the remaining 499 are primarily from Iran, Afghanistan, and further west, not in mainland India, if you will. So this is something that we'll need to talk about. Now getting to the basics of heritage biology. So we're made out of trillions of cells. All of you know what that is. There are only two components that we want to focus on. And the one is the Y haplogroup. The Y, we have each one of our cells, except a few specialized cells such as red blood cells, harbor 23 pairs of chromosomes, out of which 22 are out of them. One of these known as sex chromosome, sort of in a loose language, that consists of a pair, either XX or XY. This is high school biology, by the way. And if you had XX, you're a female. If you have XY, you're a male, right? That is the determination. X determines what sex that baby will become. Now, the Y chromosome is a very large molecule. It harbors something like about 70 to 200 genes that we won't get into that. And it is passed on from father to son, only from father to son. And because the Y chromosome is absent in a female. On the other hand, the female women contain what is known as mitochondrial. These are little power plants within every cell. They vary from a few hundred to a few thousand, depending upon that particular cell's demand for energy. Like muscle cells have thousands of mitochondrions. And it's a much smaller molecule. It's only about 17 kilobase pairs. That's the size of the molecule compared to something in the orders of about 50 or 59 megabase pairs, that is million base pairs for the Y chromosome. And it's a much smaller one, and it harbors a smaller number of genes. And that is passed only from mother to children, both male and female. And therefore, by tracking the mitochondrial gene of a particular person, you can track the maternal lineage. And by tracking the Y chromosome, you can track the male lineage, if you will. Now, that is the basic two parameters that we measure whenever we have a person sample. For example, each one of you can give a sample, and then there are laboratories in the country, I'm sure it's available in India, where they will tell you what Y haplogroup you belong to and what mitochondrial haplogroup you belong to. And that will tell you what your identity, genetic identity is, if you will. And it's become very commercial in this country over the last 10 years. There are many companies that do this, many in Europe as well. I understand there is a company trying to start this business in India, something you may want to consider looking into. Now, the Y chromosome is a very large molecule. The 60,000 base pairs in mitochondrial DNA is a much smaller one compared to 59 million base pairs in Y. And changes occur, mutations occur. Mutation is a biological term to say change. And the change, it does not occur frequently. It occurs in timescales of thousands of years. In the case of Y chromosome and the mitochondrial chromosome, it's a shorter time scale. It's because it's a smaller molecule. And therefore, there are many, many mitochondrial DNAs. And of course, there are far fewer Y haplogroups, at least as of we know. In 10 years from now, the number will expand because we'll have a greater body of knowledge. We are still discovering new haplogroups and new variants in mitochondrial DNA. And so the map of these heritage maps are subjected to change. And many of the papers published in the last 15 years will have nomenclature, which could be very confusing. That's because they get redefined, if you will. And so something that one has to be careful about. So the bottom line is there are two metrics that determines one's genetic heritage. The kind of Y chromosome you have and the kind of mitochondrial DNA you have. One comes from the mother, the latter comes from the mother, the other one comes from the father, and comes from the grandfather, and the great-grandfather, and so on, all the way up to 60, 70,000 years ago. Anyway, a little bit of a history on this discovery of the haplogroups. Although people were dabbling in genetics for heritage in the 80s and 90s, they really do not take off until the 90s. And early work in this area, if you read papers, they will look at particular class of molecules that are present in a chromosome, commonly known as STR and SNPs. Let's not get into that definition. But later on, we have now, because of availability of automation, we can examine the entire genome and look at much finer details of what is contained in genome. In the US, for $100, you can send your sample and you can get a complete map of your chromosome, all 23 and the X and Y, And you can then deal with that, comparing with different databases and compare for changes or similarities, etc. So that is now available for everyone, which wasn't the case 10 years ago, by the way. So because of this evolution of new definitions that was occurring, there was a major association organization formed known as ISOG, which is the International Society of Genetic Genealogy, which began giving nomenclature. If you're in chemistry, you'd recognize periodic table evolved during the 18th century, and they had blanks, and then they were filling it up as we went all the way to the 19th, 20th century. And so the names were given were different. If you go back to early literature, they were called by different names. And so we are sort of in a manner of speaking, periodic table of genealogy. That table hasn't been fully written. It is still being written. Keep that at the back of your mind. The key thing to remember is the data on genetic information of people, trying to speculate on where they came from and all of that, prior to 2010 would have speculations. The reason is they didn't have access to ancient genomic data. They had only current data. So that they were handicapped, as you will. So if you read papers that were published prior to 2015 or 2012, you have to read that with a pinch of salt and have to reinterpret that in the context of body of knowledge that has evolved just in the last 7-8 years. And because I showed that exploding body of knowledge in the ancient genome, if you will, that's the background. Now, this is a very crude map published in 2013. The Y haplogroup is classified as A, B, C, D, etc. Generally, A means the earliest, B is later, C is later, and so on and so forth, all the way up to R. R is considered something like 5,000 or 7,000 BCE. And by the way, this is the rough map that was generated in 2013. This has gone through some refinement. As far as India is concerned, the one that we would be interested in is in this blue category, because that's where we see L, H, and a little bit of R, and of course, a little bit of J, etc. By the way, this map is not accurate. It's a very rough map. But we do see C in India, F, and of course, H, J, L, and quite a bit of L, and of course, fair amount of R too. And of course, the map is given here that the earliest, very earliest Africans, they never left Africa, where they had an orange complex. They were not dark in complexion, they had an orange complex. Remember, we have, these are, even the people with haplogroup A living in Africa have gone through some mutation. So that is something that, and then later they became darker in complexion and they left India, they left Africa. Some of them who went to Andaman Islands, they look like this, and I have pictures of them. Andamanese have very strong African features, if you will. And of course, the C went all the way to Australia. They look like this. And of course, we have residues and ancient relatives of these individuals in Madurai district, if you will. And of course, the various other populations are located here. This is simply to give an overview. I want to give you one historical example. This would be of interest. This gentleman by name was discovered by, sorry, it hides my screen. Andiappan, I think, near Theni district. This is how he looks like. He was discovered in 2001 by Pichayappan. Oh, I could move this. So I have a helper here telling me because it was hiding the top where it has the reference to what I was wanted to look at. Oh, I do have, I could use annotate. Okay. Can you see my arrow, Bama?
Oh, yeah, I could.
Okay.
Could use
This-
a laser point.
You can see that, right?
Yeah, we can see.
Oh, yeah,
Okay.
yes, yes.
Virumandi is near Theni district. He harbors the haplogroup C M130. His ancestors were one of the earliest who came into India about 70,000 years ago. And he was discovered by Professor Pitchapin and Spencer Wells in 2001. It was a major pivotal discovery because that established the trajectory of ancient man from Africa all the way to Australia. And in Australian Aborigines, the same haplogroup is found in abundance, by the way. In some respect, the people in Australia, the Aborigines, are really ancestors of ancient Tamils, and they are related to this gentleman that I show here. And this haplogroup C has got many subclades, meaning there are various different versions of them. And they're found in India, particularly in South India, both in lower caste as well as upper caste. It does not distinguish in caste. There are enough people of upper caste who have a haplogroup, not substantial, but there's a statistically measurable number. And within India, they seem to be present only in southern part of India. Why that is the case, that's an open question. They're more present in much farther up in the northern part of in Russian region. By the way, these shaded graphs that are published by the article that I referred here is in 2013, is really in population dynamics software that generates these curves. They're not based on hard data. We have to be careful about that. But these are predictions, not actual data. But we have actual data of C being present in southern India, and I'll share some of the data as we go through this talk. Let me move on to the next slide. Oops. Okay. Oh, I did something else. Yeah.
Yeah, go ahead and share again.
Now sharing. Yeah. Okay, small technical glitch because okay. Let me get on to the next slide, which is okay. Got it. This is about mitochondrial, which is the entity that comes through the female. And this is a rough map. Again, this is an early publication, 2015. This has been far greatly refined in recent times. It approximately gives you all of the by the way, the haplogroups here starts with L0, L1, L2, L3. And then it is the L3 who migrate out of Africa into the Middle East, if you will, and then on to Europe later on. And then their mutants which has been labeled as M, they seem to come to India. And the gray one, the blue ones are haplogroup R and also N. They also have presence in India. At the present moment, the haplogroup M is considered intrinsic to India. It actually came into being in India. So if you're of haplogroup M, that means you belong to an ancient migration people into India. And substantial part of India are really fall into the category, fall into M and R and a little bit of U. And although there were many male haplogroups came into India, there are only very few female haplogroups in India. So that means most of the people who came into India appear to have been men rather than women. That, again, is an opinion based on my reading, and some scholars have shared that view too. Let me go on to the next slide to look at an example. This is a study that was done under the auspices of what is known as a 1,000 Genome Project. And I bring this up because this was done at, this particular work was done at Georgia Tech, and I live about 20 miles from where that is located. And they took samples from all over the world, but here I'm just highlighting the data that they collected about Indians. And they have used the abbreviation Bengalis in Bangladesh and Gujaratis who live in Houston, about 100 samples, and Indian Telugus who live in UK, and Punjabis in Lahore, and Sri Lankan Tamils in UK. There were approximately 500, and they had a total of several thousand that they looked at, if you will, although it was under the auspices of 1,000 genome projects. And now these are the various population groups, the five population groups I mentioned. And you'll notice most of them, although this goes from Pakistan all the way to Sri Lanka and to Gujarat, of course, you find most of them belong to the haplogroup M, R, and a smaller portion U. M dominates. So this is an important characteristic of Indian population in general. And scholars would say this was a result of Indian females who lived in India long time ago, and not females who came into India later, 2,000 or 3,000 years ago, or even 1,000 years ago. So that's an important point to remember. U is a subclade of M. M is sort of the mother. From there, it germinates N, R, and U. So they're related, if you will, in terms of genealogy, but one descends from the other. I'm throwing a lot of nomenclature at you, but unfortunately, there are no shortcuts to this. Okay, let me take a historical example now. Remember, we just did a Y haplogroup and then a historical example in Y haplogroup. Now we're taking a mitochondrial haplogroup, and we're looking at a historical example in this category. So this came out of 4 genomes that were excavated in mid-Euphrates Valley. And they found they had mitochondrial DNA that was M, various versions of M. And of course, in Syria, there are no females with no M lineage. So they were open to question. And of course, there were some Indian scholars who jumped onto this. In fact, this work was actually done in China, if I remember right. No, this was not in China, the later one. Vital et al., the full reference will tell you, they took it up and looked at the genome population and said this was, they labeled it, and one was this M really belonged to the subclades of M were found in elsewhere in the eastern part of India, and one was found in Dindikal in southern part of India. The details were done by the group that was in China, as I remember, right? Yeah, this is a work that was done in China under Professor Zhang's laboratory. And of course, there were several Indians on that group. Publication was 2014. The ones that I've highlighted yellow are the ancient Mesopotamian data. They were of M49 and M65 and did their subclades. What they discovered by looking at over 15,000 samples of mitochondrial DNA, 11,000 from the literature and 5,000 of their own samples, that M49 was found, its subclade, not directly M49, but its subclade in West Bengal, Myanmar, and Assam in the northeastern part of India. And the other one, M65A, was discovered as M65A in Dindikal in 4 samples that they worked at. The middle column, which is hypervariable region of it, these are markers of their methods of determining this particular variant, if you will. It's a methodology associated with characterizing that particular haplogroup, if you will. Those are the places that the variations are noted, and so they measured that part instead of doing a full sequence. And I think because it requires less time, less effort compared to doing a whole part of the mitochondrial. And I think that is probably was okay in 2014. Nowadays, we tend to do the whole thing because the time taken to do the way they approached it and the time taken to do the entire mitochondrial is pretty much the same, and the cost is nearly the same. There's no pretty much difference. When you do that, you might get the additional information. The key message to note is the time frame provided gives us information on perhaps some historical path. I don't want to get into the interpretation of this, but we know there were ancient routes from between Indus Valley and the Mesopotamian culture. There are strong evidences for that. And there were also evidences of trade between southern part of India and folks in Rome, which would have gone through the area which is near the Arabian Peninsula. So there's some connections there too. So what the trajectory of human pathway on this is of course up to interpretation and speculation, but hard data cannot be debated. Data is data, right? I've always told my students, data do not lie. It is the interpretation that could fall into the wrong interpretation. Now, this is a good place to stop. I've given you an overview of historical Y and historical mitochondrial DNA. And now, given you what the two major markers are, I'll open up for questions and discussion before we move on to Indus Valley data that's available. Thank you.
Thank you.
Questions?
Raj, I have a question. You said that the mitochondrial DNA separately can be sequenced along with the nuclear DNA. So both are available now in the United States or anywhere else?
Yes, it is. Like if you send a sample to 23andMe, which one of the agencies that I've used, they will send you not only the what haplogroup that it is characterized according to ISO, which is the sort of the gold standard, and that information will come to you, but you'll also be able to get the entire chromosomal data fragments, and you can download it, and so you can get both the information from them. So, and there are many agencies that do that. Many commercial operators do that.
You said that females in India, so it's more ancient, and the M is there, M group. So can you comment on that? How long ago they were, can be dated back their presence, and how late the male moving in?
Well, this is Spencer Wells' words, I think, sort of. I'm summarizing it. And also it's been said by many other people too subsequently that the ancients who came in did come with their females. That is, they moved as a family, it appears to be, because the M is sort of has dated to be 60 to 70,000 years ago. And that is approximately coincides the onset of entry of humans from Africa into the Indian peninsula. And the residue, for example, Burmandi, CM130, being near Thani district, authenticates that presence. But we also have very strong evidence of the Andamanese, who belong to haplogroup D, which is a later variations of C, and they are intact. And they were an isolated community. And that haplogroup tells us, and that piece of data is extremely important because that work comes in the next section of my talk. And so 60 to 70,000 years ago is something to keep at the back of our mind. And remember, they continue to multiply as they went on to Australia. So my speculation is they moved as a family. And according to Spencer Wells, they walked along the coastal route all the way across. And this is pre-Ice Age. And they had to cross only a 70-mile stretch on sea. Everything else was on land because all the islands in Indonesia were connected at that point in time. You remember, Ice Age ended around 12,000 years ago. So this is pre-Ice Age. And so their residues are related to the Thani district gentleman.
Thank you.
Dr. Butterson, I have a question.
Sure.
For the female, the X and X, one comes from the male's mother, right? And then, so how do you distinguish between the female's mother and the male's mother. Do you follow what I'm trying to say?
Oh, yeah. That's an excellent question. That comes in a later slide all the way at the end. So I will address that as we get there. Is that okay?
I have a question, Aya.
Yeah, go ahead.
The people in the mountains, the Todas and the Kurumbars, all those people, they are all the original descendants from Africa?
Well, that slide comes a little bit later. You may want to wait a bit.
Okay, thank you. And another question quickly.
Sure.
The Aryans came through the Khyber-Poland bypass, and they did not bring any women. That's what the historical books say. So they came and mingled with all the Dravidian women, and then we are all the descendants of this mixture.
That is exactly correct, but that includes in the next section of my talk.
Okay, thank you.
We can go on next section.
Yeah, this is Guna from Fremont. I just wanted to have one question though. Thanks, Dr. Rajaya, for explaining such a complex subject to layman terms though. My question is on the topic heading itself though. So nowadays, the notion and identification of Tamils are hotly debated politically, culturally, and scientifically though. The question is, generically, how do you define who are Tamils from scientific point of view? I believe that the publication of such a study and scientific analysis will pave a way to clear an identity issue that emanates in and around India and the world for political and cultural identification purposes.
Okay. It's a very good question. I want to give a short answer because we could get into that in the last section. The important thing to remember is the language you speak now and your genetic heritage are not necessarily connected. And so when you say Tamils, if you're from my point of view, if you speak Tamil and that's your mother tongue, you're a Tamil. I mean, that is sort of the way. And your heritage is all over the map. It is not single. So there is no pure Tamil from a scientific point of view. There is a Tamil who is of haplogroup L, who's of haplogroup C, who's of haplogroup R, R2, which is native to India, by the way, or R1A1, which is the so-called Aryan genetic heritage, and so on and so forth. So we have a mixture, if you will, a blend. But the heritage can be deciphered, but the particular question about pure Tamil and pure something does not really exist in the real world. It exists only in common man's language, if you will. But if you're a person who speaks Tamil in a simple way, you're a person of Tamil, and you may not be of Tamil descent. For example, the third generation Indians in this country, many of them, for example, of Tamils, they know they are Tamils, but they do not know how to speak in Tamil, and they can understand a little bit of Tamil. They speak only in English. And of course, if you look at the genetic makeup, they would be very much Tamil genetics, right? So that distinction needs to be kept in mind. And I have a slide that will articulate this very nicely for you later on. Okay?
Thank you.
Yeah, we are short of time. So maybe we take one more question and move to the next one. Uma, you had a question?
Yeah, I had the similar question. It was answered. I would like to ask that one question about the haplogroup that Aya was describing. தாய் வழி சமூகம்னு சொல்றாங்களே ஐயா, is it based on that mitochondrial DNA thing that you were explaining? That is what it is, or
no, that is not what it is, I think. Okay. But in my opinion, since you raised that point, தாய் வழி சமூகம் meaning motherline is because I think this is ancient man. This is my theory, by the way, not proven. Others may have a comment. Since a woman can create a whole living being, female was given the deity status, a godly status. And that you see in the Tamil culture and in Indian culture and in Indus Valley culture. And you also see a little bit of that in other Middle Eastern cultures. So that is my explanation. I'm sure there are many, many different viewpoints on this, but that's a long debate. That's a nice discussion at a later point in time.
Thank you.
I'll move on to the next, Bama.
Yeah, please. Daisy, you might type your question in the chat box. We'll take it in your question first at the Q&A session.
Well, if we do run into short of time, we could continue this next week or something like that. But let's continue on. I'd like to get through most of it and then see how that happens. Yeah, we're almost 40 minutes into it. So, all right. The next one, this is an important piece of work that was done.
Daisy had a question. We'll take it up now.
Fine.
Yeah, Daisy has a question. So should we take it now?
No, we should, my submission is that we should let the presentation happen. Questions we can reserve for the end.
Okay.
Yeah, this is my question actually. Rakti, the sample, is it which haplogroup?
No, let's wait and see
question.
that.
Thanks. That is my question. That was my question. Thanks.
Can we wait him to make a presentation? That's what he's going to speak now.
Okay, let me continue. Okay,
please, sir.
This is a very important piece of work that came out not too long ago in 2019. This is because of the excavation of the skeletal remains. And David Reich's lab was involved in this. And in fact, all the analysis was done in his lab. Shinde is the person who heads up the Deccan College, which has a very long history of work done in archaeology in India. So it's a very prestigious institution in my view. They've done some pivotal work. The key points on this particular chart is that abstract of that paper. A very important piece of data is contained in this. So, and I've captured the most important point on the top, but I've devoted several slides on this particular topic here. Skeletal DNA from members of ancient DNA, from ancient Iranians. They show ancestry to ancient Iranians, important point. Before their adoption of farming, that is, the people who came into Indus Valley were not farmers initially. That's an important thing to remember. They didn't have that connection to the Iranian farmers. Many scholars would say Iranian farmers came into Indus Valley. That is not appears to be the case. So this pivotal discovery sort of shut down that theory. And they were from Southeastern Asian hunters-gatherers. They were hunters-gatherers. They were not city dwellers when they came in. So that is another important point. And they completely lacked, they had no evidence of steppe pastoralist ancestry, which in common man's language is the Aryan descendancy, if you will. They were not Aryans. This was previous to the entry of Aryans, and they make remarks about that as well in their paper. So these three pivotal points are important for us to capture from it. Although the paper is about 10, 12 pages long, the data is quite enormous. And the skeletal remains was collected from, has been dated based on adjoining charcoals 2800 to 2300 BC. That is the mature phase of Harappan civilization. There were earlier parts of Indus Valley civilization that have been known. Mehrahar, which is further west, is even an earlier one where they have residues of transformation from hunter-gatherer to city dweller. Very important site. Not enough work has been done in that area. And these folks were in cultural contact with Gonur and Shar-e-Shokta region, where 4 ancient genomes were excavated. That was used in this particular study, if you will. So this is David Reich's and Shinde's hypothesis. I suspect it is David Reich's because he has a long history of work in ancient genomic data. And the bifurcation, the hunter-gatherers from Iran moved into Indus Valley approximately 1000 BCE. This is long, long before. And Rakhi Gargi, there are evidences of occupation there going back to 8,000 BCE, I think. So this appears to support those early settlers in that part of India were hunter-gatherers from Iranian region of the country. Now, how do you figure out what their genomic content? So India has no ancient genomic content except this particular lady. She was a relatively young lady. And of course, they put that in this space of mature. The y-axis here is the timeline, if you will. And of course, the data they type here is 10,000 BCE. And of course, they have collection from Dr. Thandaraj's data. He was a pioneer in trying to characterize the Andamanese genome. He works at CCMB. He's done some pivotal work in this area at Hyderabad. It's a wonderful lab that I visited that lab many years ago. That I don't think I met him at that time, but I gave a talk on some other aspects. And he has a complete genome of Andamanese, and that is available as the present. And the researchers in this space examined the Andamanese and many, many other ancient genome and finally discovered the lineage that came from Iran had the closest connection in terms of similarity, if you will. So a lot of details that I'm skipping. And these residues were Zagros Mountains, which is east of Mesopotamia in Iran. And Zagros is an important element. And in the Zagros Mountains, and they combined that together to predict that the connection of the genome of this lady varied in the space between using certain models between 50 to 98%, 98% meaning totally Andamanese and 50% meaning equally split between these two genomes, right? That's a sort of a rough and tumble number. Don't take that to be precise, but that's the nature of this field, by the way. Now, how do Andamanese look like? This is an example of how they look like. And this is, I visited this about 4 or 5 years ago near Ooty, because that's where I went to high school. Tribal Research Center is just 10 kilometers from Ooty proper. They used to be near Botanical Gardens. They moved it out as expansion was taking place. It's a very nice facility. They have a very nice collection of tribals. And this is one picture of them. This is another picture of them. And here is, and this particular tribe is known as Jarawa tribe, Andamans. And of course, they also have in certain islands folks who look like Chinese. They're not. They are haplogroup O, by the way. But the Jarawa people have haplogroup D. Remember, D comes after C. C is what we found in Theni, right? So these folks came a little bit after the folks who came into India around 70,000, or probably about 50,000 years ago or so. Okay, let's look at IVC phases, Indus Valley phases. I'm just giving you a few examples. The one on the left is, if you look at it very carefully, it's got a pivotal, or rather a jutting out jaw, which is characteristic of Negroes, and a broad forehead, and a round flat nose and a broad face, as opposed to a narrow top line face, which is more what people in this space call as alpine. The second one is at Boston Museum. That's one of my pictures. It's a very tiny one, maybe about 4 or 5 centimeters, and they labeled as somebody who's throwing a spear. And look at his face. His jaw is jutting out, pretty much like the gentleman on the left-hand side. And these are faces of Indus Valley. Let's look at a few other. And the left one is in Delhi Museum, a picture I took many years ago. And the right one is from a publication of Porchal. Again, a jutted out face. And of course, a face that is broad and round is not uncommon. Tightly knit hair, for example, on the left-hand side here, this face, that here, the gentleman seemed to have mated the hair and created a sort of plaited the hair, if you will, on the top. And this is a common feature that ladies of African descent do that in this country, by the way. Maybe they discovered this style 4,000 years ago, before the American African women discovered it, only about something like about 30, 40 years ago. Anyway, interesting combination. But the key characteristic of the hair appears to be, this is my non-expert observation, the hair is thick and highly curly. And this gentleman on the right has his hair bundled up at the back like a bun, if you will. And it's very common. Folks in the South India, particularly of people who are typically would call themselves as Dravidian, have thicker hair than people who are of Aryan descent would have thinner hair. And folks in, for example, in Europe will have hair which is about a couple of microns thick, 1 or 2, as opposed to, say, thick hair people will be more like about 8, 9 microns, which is a measure of thickness, if you will. A micron is 1 millionth of a millimeter, I think. 1,000th of a millimeter. Okay. All right. These are 2 examples. Here is another one. The left-hand one is a famous dancing lady. Thick lips, thick nose, jutted out jawbone, and sort of a broad face, broad forehead, somewhat characteristic of African features, if you will, like the Andamanese we saw. The right one is also from Indus Valley civilization, which is more longish face, and of course a hair that is knotted up on top, and a shorter forehead, not a jutted forehead. So one non-expert opinion is there was a blend of people in Indus Valley civilization, and that is not uncommon. That should not be surprising. Remember, Indus Valley runs probably about 1,000 kilometers north to south and about 1,000 kilometers east to west. It's a very large area. And of course, if you travel 1,000 kilometers in India, you'll run through 2 or 3 language belts, right? Even in Tamil Nadu, you'll run into different dialects of Tamil Nadu within 500 kilometers. Okay. The highlights that that particular article points out, he points us out as a highlight of that work, is a sample verse from a population that is largest source of ancestry. Now, that is a speculation in my view. He believes that the descendants of South Indians are related, very strongly related to this particular lady that was discovered at Rakhihargi. Maybe, may not be the case, but that is an opinion, not a stated fact, because they didn't determine haplogroup. They just looked at only the genome. I do not know why the mitochondrial DNA haplogroup was not published. Of course, she wouldn't have had a Y, but that was not available, at least in the publication. I haven't written an email to David Reich about that. Maybe I should do that. By the way, David Reich is very prompt. If you send him an email about a question about his paper, you'll get a response within a day. Either he does it or his postdoc does it. He's very prompt at responding to questions. So that's very helpful. He's very collaborative, by the way. If you have questions, he responds very nicely. The second most important point, I pointed this out with the first slide, the bifurcation between the Iranian hunter-gatherers and who then migrated into the Indus Valley, because the genomic content of that lady exhibits that early hunter-gatherers in Iranian Peninsula and Zagros Mountains. And finally, very important, there was no genomic content that related to the Fertile Crescent, meaning the ancient classical people who are to have invented farming. There was no genomic content of those people in the so-called Mesopotamia, all the way up to Levantine and all of that area. And so that seemed to speculate onwards to the folks in Indus Valley discovered farming on their own, by their own invention, if you will. That possibility exists, not a proven fact. That's just an observation, if you will. And now the authors, Shinde and David Reich and his co-workers, get on to the linguistic domain. The first important one they mention is ancient South Asian farmers, meaning folks in India, represented in IVC clan, meaning the folks in Zagros, had negligible ancient and Anatolian farmers, and as well as Iranian-related ancestry, but they only had the hunter-gatherers. So they were early people. They were very early people, not people who acquired all of these technologies and then came to IVC, Indus Valley. They actually appear to have invented everything that they had on their own, perhaps through cultural diffusion, but they did not, the genome of that lady did not point to the fact that they were migrants from that culturally rich area. That is the important point to remember. Second one is language spreads. This is a theory among the linguists. Language spreads during pre-state societies, that is before you have settlements, by large-scale movement of people. Large number of people have to move for the language to move with them. Here, that is not the case. And that is an important point to keep in mind. And then a very important point is, because of this particular set of data, and he has done, by the way, a lot of data on ancient DNA with steppe pastoralists. So he says the natural route for Indo-European languages to have spread into South Asia, that is India, is from Eastern Europe via Central Asia, that is north of, from Poland into steppes, which is current Ukraine, in the first half of second millennium BCE, a chain of transmission. This is now well documented in ancient DNA. So the pathway was not directly, there was a theory that they moved into Iran, they mixed with Iranians, and then moved into Indus Valley and then into India. That is not the case according to this particular article. They moved from Poland area to steppes region, and then that is documented using ancient genomic DNA. And all of that body of knowledge came out only in the last 6-7 years, by the way. And then finally, which is, I think, something I've said, the fact that steppe pastoralist ancestry in South Asia matches that in Bronze Age East Europeans, but not West Europe, provides additional support. He has several publications in this area, published around 2016-17, because he was a pioneer or a major player in the ancient genomic data. And so he had access to this and was able to provide this perspective. This linguistic perspective is an important part because it tracks the trajectory of the incoming folks, right? And finally, now this is a further gradient. He also says with just one piece of data, we shouldn't draw a lot of conclusion. So this is like saying, I've said all of this, now I should be careful. So in his last paragraph in that article, they say that the gradient, that means it's a prediction based on 12 samples and one in Indus Valley. We shouldn't say a lot. So he's being a little apologetic for the conclusions he draw. And that is the mark of a good scientist, right? Because you should have strong evidences to go forward. And then finally thing is, a lot of work needs to be done. And I think I mentioned this earlier. We don't have a lot of ancient DNA data from India, except for this one lady. Only one piece of data we have. We have a ton of ancient bones available. There's a great deal of technology that has come to the fore in the last 10, 15 years. And that needs to be harvested. Folks in India really need to pay attention to this. I think that should bring up my break, I think. No. Okay. The second important point is haplogroup L. There's been a lot of discussion during between 2005, maybe even earlier, that the folks who lived in Indus Valley belonged to haplogroup L. We don't have any evidence for that at the moment, by the way. Remember, we have only one female child and a child, I think, also a female from Indus Valley, not a male yet, not yet, although they have collected them, but they haven't yet been analyzed. So this publication goes back to 2013, before ancient DNA, but there has been a speculation among scholars based on indirect data, based on what's available in haplogroup L in the Indus IVC Klein, which is in the Zagros Mountains, because many of them belong to haplogroup L, that the folks in Indus Valley might be haplogroup. That thought process has been there. And they had speculated based on that piece of data, this chart that's in Wiki, but it's not a fact, by the way. And this number is also overblown in my view, but nevertheless, and the chart of how haplogroup L occurs is given in Wikipedia. It's been considerably expanded by a recent publication that came out just 4 months ago. And I posted it, I think, in the JOV group, I think. Let me get on to that. This was published in June of this year. The title is a little conclusive, little optimistic. Human Y chromosome haplogroup L1M22. That's a subclade of major haplogroup L. Traces Neolithic, meaning when people began to work with fine stone tools, that is an early settlement time in West Asia, and supports Elamite-Dravidian connection. Those two key terms really caught my attention. And I have a lot to speak about it, but I'm going to limit myself to the genetic aspects of this. So based on that, what they say in the paper is that the origin, they rely on ancient DNA, by the way, is the origin of haplogroup L, one particular subclade of L, L1M22, is this region, which includes Mesopotamia, and it includes Elam, and Zagros Mountains is somewhere here, Indus Valley is somewhere here, so they are neighbors, if you will, originated here, and it expanded into West Asia, but also into South Asia about 8,000 years ago. And the evidence from Rakigargi data on the female seem to support this, because it says it's coming from Iran about 10,000 years ago. This gentleman, these authors say it's 8,000 years ago. In this space, 1,000 years apart is approximately the same. So, and then they expanded into India 4,000 years ago. That's 2000 BCE. This is a map. And of course, this is simply a model, not based on hard data. They then rely on many thousands of samples. They took 165 ancient DNA samples picked from various regions, and they sampled taking data from various different places. And those locations of samples are noted here. There were different sizes. By the way, this was done this year. That means the work was done last year. By this time, they have a huge database of current day folks in various parts of the world. And also they have access to 10,000 ancient DNA databases. That was not available to Shinde and Reich earlier on when they published that work in 2019. That's just a matter of 5 years. So the expansion of new knowledge, new data in this space is very impactful. And then they predict, these are predictions, that the points that they use for predicting the shaded map is based on the genetic content of people where they took their samples from. Which includes the black spots. I won't get into the detail. And these are different haplogroups, if you will. I want to keep in mind for us LM20 and LM27, because Spencer Wells had speculated all the way back in 2005 that the folks in Indus Valley were of this haplogroup, LM20. He did not have data, but he had indirect information, if you will, based on early haplogroup classification. That was about nearly 20 years ago. And of course, the later variant of the descendant of LM20 is LM27, and it seemed to harbor, based on finer genetic analysis, another mutant known as M76. These two are important markers, and because where they predict, based on their data, Rajasthan, if you will, and also in Kashmir, and here again, Rajasthan, these haplogroups, and a little bit in South India, more closer to the region that many of us come from. So I'll go from there to the data. Now we are looking at hard data. The y-axis is really different versions of this haplogroup. We don't need to worry too much about the various nomenclature, but the horizontal axis is very important. It tells you the years before. So 0 means current. The horizontal various values, all these labels that you see here, are current day people's data that have become available to these authors. They were dealing with a huge number of data that has become available. By the way, there are certain protocols they follow when they use data for research, and I'm very well aware of it because I was using human samples and I had to sign all kinds of guarantees not to disclose identities, etc. So these are unidentified biological samples of current day people in various different parts of the world. That is what these labels are. And L haplogroup, this mother group, if you will, mutates to this M27, I'll use a SOB abbreviation, and of course, the various different versions. And this took place, according to this author, approximately 8,000 years ago. And that is based on hard evidence of a sample from Zagros Mountains. That's what this black dot here means. And similarly, this further variations of that is here. And that took place approximately 7,000 years ago from today, and various different ones. All of this, bulk of this are from the Iranian region or Afghanistan region, none from India, by the way, and all the way through to this end. And these are descendants of these folks who lived in Iran and Afghanistan, etc. And look at their names. These folks are Brahui speakers, Sri Lankan Tamils, Punjabis, Telugu speakers, and Sri Lankan Tamils, and various different people in Singapore, Tamil-speaking Indians, classified as Indians in Singapore, and Pathans in Pakistan, and a whole series of them. Okay, and a lot of Gujaratis, by the way, interestingly. And remember, Gujarat is not far from where Indus Valley is. Let me go on to the next slide. Haplogroup L. Now, this publication 2017, so before rich data came into being, and these authors in Europe looked at a particular class of Indian population, the Jat community, and they took 302 samples of Jat community, both in India and in Pakistan, because they're culturally, they are continuous, and the language is also continuous, by the way. And apparently there are about over 120 million Jats, both, when you combine both the population in India and Pakistan. They did not do full classification of L because they were probably short of time or energy. They just looked for what is known as a molecular marker that identifies the genetic heritage as being L. So it's a limited investigation, nevertheless very useful. But of course, if they had published what particular one is, we can go back. I have not done that. Look back and see whether we can classify them under which one it was. But that doesn't matter. 1/3 of the Jat community in Rajasthan and in Pakistan belong to haplogroup L. Now, that's an important indication. Now, when you look at it in the context of Spencer Wells' hypothesis, an earlier hypothesis by many scholars, not only those, that the Indus Valley was done by L, that could be, L was one of them, but not necessarily all one, was an important one. They also examined Jat population for these various others, such as R, etc. These are important classifications. I'll speak a little bit about them later. And I think that should bring us to a break. So I've covered what Shinde's and Reich's work talked about IVC, and what...
For the, let's continue with your presentation. Maybe we can take the question answer session at the end of your presentation.
That would be nice, I think, to try to go for the presentation.
Okay, let me continue on.
How long you think, Raj, next segment?
How long?
This is the final segment.
Another 20 minutes or so? If it's too long, we can have another talk.
Just another 20 hours.
Okay.
No, I could speed it up. Remember, a professor starts his talk and finishes in 50 minutes, right? Sure. So,
phenomenal.
So it's up to you. You decide whether to cut it or move on to the another day, another lecture.
So I'm used to this clue students give, but packing their bags and looking around left and right. But here I don't have that feedback. So my time is determined by my interest, not by the interest of my audience, right? Wait, let me go forward.
Okay.
I'll try to speed it up. Okay, this is a work that Professor Pitchayappan, along with Spencer Wells, is involved as an author. This came out 2012, long before the ancient genomic data became available. They chose samples only in Tamil Nadu, and his students, by the way, have published a very nice thesis on sampling at various parts of India. That's, by the way, that is downloadable. It has rich data, something that might be useful in a future talk. Nevertheless, they looked at why they chose those spots. We weren't told. We're not told in the article, but they seem to have chosen a lot more tribes and Adivasis than the normal population. They did include normal population, but only a minority. Nevertheless, they draw conclusions which are sweeping conclusions, which I don't quite agree with. Nevertheless, they give various samples as to where they took them and all of that. I'll fast through that. The key message from this is they found about the tribes. The first one is hill tribes, another hill tribe, and scheduled castes and farmers. They found a good number of them in F. By the way, F is not as well studied as L is or R is. And so we are in a domain which is not as well studied because there seem to be far more of them in India than in elsewhere. So since Indians are not engaged in this research as much as the scholars from the West, and so we are sort of in a little knowledge gap here. So the hill tribes who are foragers, meaning as opposed to hill tribes who cremate, seem to have a fair number of F, small amount of 10%, 8% of C, the gentleman from Thane district, and they do have R2, R1A, which is a so-called Aryan gene too, 1% of Aryan gene. And you will find that to be the case. For example, the hill tribe near the Thodas and Kotas, they have a much higher R1A content, the so-called Aryan genetic marker. And of course, you will find this distribution in a very short way, in a short term. We are a blend of cultures, blend of genetic heritages, not pure in a sense. But there are certain things that are clearly absent too, if you dig into this table. I'll just go on to the further ones. For example, the upper castes have a higher percentage of R1A1, which is about 8%, compared to in Tamil Nadu, typically reported as 2 or 3%, but it's higher. But they also harbor C at level of, in this particular sample, it's in the orders of about 10%, by the way. There are many other studies that seem to indicate they also have E. This is a very unusual, typically considered Ethiopian, if you will, E, in a sort of a crude way. And of course, they do have L as well in the very small percentages. I won't get into the details of R for now. And now they speculate that these R1, A1, M17, L1, M27, and so on were autochthon ones, meaning they originated in Tamil Nadu. They seem to claim that, but of course they didn't have access to ancient genomic DNA that was available to the previous author. And therefore they drew this conclusion based on their limited available information. Of course, this sentence will not hold true anymore. The L1 M27 came from Zagros Mountains. It is not native to India. Those folks came into India is what we know. And the other one is R1A1 has nothing to do with native Tamil Nadu. It actually came into Tamil Nadu around 1800 BCE. Let me go on to another study that was published not too long ago, 2008. Again, they didn't have access to ancient. Here I'm only looking at Y haplogroup. I'm not looking at the features of migration. They classified various different kinds of people. We don't need to get into that. The last column is what we want to focus on. And the data I've summarized on top in the block is haplogroup is found among Tamils at about 3% level, according to them, compared to earlier study of Pichayappan, 0.3%, right? So it's a little bit different. So sampling has a great deal to do with the numbers you come up with. Haplogroup F and L is 13, which is more linked to Indus Valley. H 20%, R1A1 26%. That is rather high. And R2, which is thought to be native to India, 10%. More discussions to be done about R2, but I won't get into that particular topic today. And for mitochondrial DNA group, it's primarily 2, M and N. N is a descendant of M, by the way. So all these ladies who belong to various different castes came from the native Indian females, if you will. That's what this is. Although there are stray points of foreign, but most of them seem to have been native. There are small changes, by the way. So this is a question and answer session, which I'm going to skip. Now I'm on to my DNA journey, close to the last part. So what I did was I had participated with the National Geographic they classified me as L20 because that was a body of knowledge that existed at the time. This was done a few years ago upon the insistence of my daughter-in-law, who had done this. She was into this field. And she found, they found me to belong to LM27. I think I mentioned this, and my ancestors of this are in Zagros Mountains from approximately 8,000 years ago, right? Now, that is one aspect of it. The second, in terms of the mitochondrial DNA, I seem to belong to R, which is a descendant of M. From M, you get N. From N, you get R. And these are all native to India, various different variations of them. The key interesting aspect are, I've highlighted them to say 1 or 2 minutes about them. The first one listed is my son's name, and he has 22 segments. That is, what they do is different segments of the chromosome is tested. And of course, they didn't know that he was my son. The computers discovered he was my son, and they reported it like so. So we could beginning in computers as the software aspects of it, right? And of course, there are people that are listed, which I was in contact with one of them who lives in Brisbane, and he was of an Australian descent who happens to be the husband of Manonmani, who is a daughter of Nadarajan and Punidam, who migrated from the same village where my grandparents came from. So there is a connection. The fact that there are 4 and 5 segments connection means he's really a closely related via some marriage. I have no idea. And he, being an Australian, he was very interested. So we had a couple of email exchanges. It was an interesting one. Their daughter is Sorna, who's doing his nursing at Tasmania in Australia. I had an interesting exchange of information. For Prakash, he lives in Amsterdam. His grandparents came from Brookspet. In Tamil, you'll call it Purgesapattai, as my grandmother used to say. It's a small little village just outside of Kadalur. And that is the village where my great-grandfather came from. And so there is some connection with there. And I share one segment with him, meaning there is some close connection. He's not very far genetically, if you will. He's a blood relative, or rather direct descendant relative, but I don't know who he is via whoever I know. The more interesting ones are Sumaya Khan. She's from Karachi area. And Yatrik Patel, he's from Gujarat, as the name would suggest. And 2 others I just want to highlight. Mahendra Patel, he's not related to other Patel, but he lives in North Carolina, but he's from Gujarat. And then Anusha Charania, she's from Karachi. And they're all related by sizes of fairly significant chunk of chromosomal DNA. That's an important part, which means there's a close relationship, but it's not direct lineage. So from these pieces of my own experience, I have 2 lineages into Pakistan, Karachi, and 2 in Gujarat, 1 from Maharashtra, 1 from Hyderabad, several in Tamil Nadu, and I have lineages in Fiji, Trinidad, and Vietnam. The gentleman in Fiji is migrated from Kadalur area. He does not know which part, but he's 4th or 5th generation, but he's related. And Trinidad also from South Arcot district, but he doesn't know where he's from. We have had 1 or 2 email exchanges. The Vietnam story is an interesting story, but I won't get into that now. There is a possibility that my ancestors' trajectory was Indus region to Tamil Nadu via Gujarat and Maharashtra by the relatives that I seem to have discovered through 23andMe. And my final remarks. Now, there are needs in additional heritage markers. As I mentioned, the 3rd leg of genetic relationship is the genes, which is really what does the work in your body. And we have approximately 20 to 25,000 genes in every one of us. And genes contain 2 parts, those that are coded, those that are not coded. So only a small % of it is coded. And they carry signatures. And some of the genes are conserved, they don't change like that. Genes associated with the hemoglobin is conserved, they don't change. All the way going back to the monkeys, they have very small changes. But the other genes go through mutations, the non-effectively useful one. For example, the genes associated with the skin of your color goes through mutations based on your environmental conditions. Most people have light skin is because their genes are still working to convert their the so-called SLT1 gene to make them more Malpighian layer, right? The color that's associated with the color. So these adaptive mechanisms have been come to the fore in the last 5-10 years, and they provide rich information for trying to locate genetic connections. This is a quick example. Square is a male, circle is a female. I've artificially assigned a haplogroup L and M and the generation 1. They have 3 children, 2 males and one male and 2 females. And the females will inherit the same haplogroup as their mother, M. And of course, the male will inherit the same haplogroup L as the father. And the female, say, marries a haplogroup H, and they have 2 males and 1 female, and that would be M, H and H because of the male-to-male connection. And say the male marries a female who's of U, and then they have children, they would be H and U, and these haplogroups will have nothing to do with generation 1 haplogroups, but they will share fractions of the original chromosome. Since chromosomes are divvied up with the children, they would have 1/8 of the chromosome of each of the parents. And that is what they call segments in 23andMe. When you have one segment out of 22 segments, they're related by 1 divided by 22 in terms of generations, if you will. So there are nice calculations one can do based on information. And also the segments you test for can be made long, short, to look for near relatives and far away relatives, if you will. Now, finally, this question came about during the first session. Man has X and Y, the female has XX. When they inherit, the female gets 1X from the female and 1X from the male. And the male gets X only from the female. He does not get it from the male. And the Y comes from, of course, the parents, from the male. So the X component in the male contains genetic information associated with the female also. Now this area, but X has a very interesting property known as recombination. And so it is not easy to decipher. There are a lot of researchers working in this space. That aspect may add another dimension to understanding genetic linealogy. That is in the far into the future. This is at a very early stage, by the way. And so I just want to highlight that. And then finally to point out, if you're ever interested in any of the haplogroups, you could go to ISO's website. I've listed at the bottom. You can divvy it up, you can break it up and download the entire Excel spreadsheet and look at chromosomal data of yours against that and do all kinds of analysis, if you will. That brings to the end questions. Okay.
Thank you, Aya. Thank you for this.
Fantastic talk, Raj. Excellent data, and we don't have time. That you have very brilliantly articulated with illustrations. My quick questions is, Indus Valley, you said possibly they have evolved agriculture in parallel. That how much data is to support that? And compared to the Y chromosome and mitochondria, which is more reliable? Looks like when he marries a different person and that mitochondrial DNA is moved away from the lineage. So
okay.
It's like Y chromosome is more stable. That's it. Thank you.
Well, I'll answer the second question first. The Y and mitochondrial DNA, it's almost like asking which eye is more important, left eye or right eye? So although you can see about 160, you really see a lot more with 2 eyes, right? So both are important. The fact is, mitochondrial DNA is smaller, and therefore being able to characterize it is very quick. You can do it very quickly, overnight, if you have one of those machines that Applied Biosystems manufactures. So the issue of length is no more. You're absolutely right. The mutation that occurred in mitochondrial DNA is more than occurs in Y. Therefore, the dimension of genealogy is greater, and therefore you need to look at it more carefully, if you will. But that data is available. The data intensity of data is no more an issue because I can solve simultaneous partial differential equations on this equation in this device now, while my fellow PhD student worked on a mainframe for 4 years to solve the same problem, right? So the availability of the technology has really oversimplified many of these complexities. Okay. I hope I've answered your question.
Yeah. About the Indus Valley agricultural parallel
Okay.
evolution.
The question about the premise on which Professor Reich and Shinde argue is, since the early settlers, the settlers at mature phase, that's an important term, mature phase of Harappa did not have the genes associated with the later farmers of Iran. Therefore, those people at that time had already invented farming. And therefore, those folks had that capability and they must have developed it in situ rather than brought it with them. You understand that logic?
Okay. Yeah.
That is the way their thinking has been.
Sure. Thank you.
Daisy,
So Chale, can I just
thanks,
for
Baba.
1 minute?
டாக்டர் இவங்க பேசுறாங்க டாக்டர் பாலகிருஷ்ணன்
பாலாஜியா பேசுறாங்க.
பேசுங்க.
Thank you very much, Dr. Raj Muttarasan. When we met, that very one day meeting, we could understand how you are going into that. This is a very eye-opener for us. And the polemics apart, very important thing, you mentioned that the number of data available, like ancient DNA data, which is flowing from all over the world, particularly South Asian, other thing, but in India only one. So this is being controlled in such a way that our own understanding that the access to the DNA material is very much limited. You may enlighten us. And second thing, even we have a feeling, for example, Adichanallur, one full skeleton was taken when Satyamurthy did a research. Then it was taken to Mysore by the anthropologists. Even today, we do not know what was done with that. And second thing also, even the scholars whom we respect, they've done a phenomenal work. But then when it comes to the presenting, in science paper, they write something. In the press conference, they speak something else. So I had an opportunity to speak to Mr. Vagesh Narasimhan, who was also part of this kind of studies. I asked this question, why Vasant Sinde's presentation in a press conference was different from what he was a co-author in a scientific paper. So there's a lot of things are there. As you rightly pointed out, we are in the beginning to understand. And that is the reason my submission is that in the JOC also, I've been talking about rainforest pluralism of India, not only current India, even Indus Valley civilization had a rainforest kind of lot of people in the small groups speaking. Thanks a lot. I don't want to take much of time. Other people will eliminate. Thank you very much. Thank you very much.
I'll just make one remark. I met both Spencer Wells and Pitch Chapman in 2019 in Chicago. So at that time, the conversation led to ancient, because at that time, this was a hot topic and I was following this. I asked a question, why hasn't this been done? So, and at that time, I was told there were 4 wonderfully well-preserved skeletal remains sitting in Egmont Museum, which was excavated from Adichanallur around almost 100 years ago. And they had applied for requests for, apparently there's a procedure that they have to follow for analysis. And they were waiting for approval since 2005 or 6, something like that, almost about 14, 15 years. So there is a gap somewhere. And when Vidya Chandran visited our area, Philadelphia, in 2018 or 2019, he, at that time, was the commissioner of the museums. I'm a museum fan, by the way, and I visited him in 2019, I think. But when he came to Philadelphia area, I did tell him that it's no big deal to start an ancient DNA lab in India. it's almost setting like a chemistry lab. It does not require a great deal of skill. And I'm very certain, although I don't know David Reich directly, but I know from indirect interactions, if you send a couple of people to his lab as a postdoc, they'll be trained, and then you can set up for less than a few $100,000. It's not a big money. And you can do all the analysis yourself. You don't need to rely on any external help. I think one answer that came back was, I don't know how true it is, they don't believe anything that is done in Indian labs. I mean, that is a sort of a shock.
Yeah, that credibility issue is there, doctor. Apart from that, I wonder. Of course, we are not a professional like you, in the sense that people are afraid. They don't want to face the fact because there's a lot of resistance for this kind of studies in our country, and there's probably the skeletons will fall out of the cupboard. Probably people don't want to touch that subject.
But I think there is the other observation I make is it has been a tradition because of what has happened. There's a lot of IVC skeletons that are also sitting in the museum in Karachi and also at site museums in Pakistan. And so those also need to be given attention. They're not in good economic conditions to pursue that, but that is something that hopefully in the next 10 or 20 years will happen. That's my hope.
I want to add one point here. I happened to attend Professor Tangaraj's lectures, and in that lecture as well, this point was brought out, like the Sadichinallur skeleton. He mentioned that the skeletons, whatever is available currently at the Egmore Museum or the ASI one, cannot be tested. Like it cannot be used for all the genetic experiments. The condition of that skeleton samples is not so good for testing, is what he mentioned. Like that is another.
Although I have not handled ancient samples myself, the fact that the method that was developed in Copenhagen, being able to extract from, apparently the molar is they had only one single tooth to extract and evaluate from multiple sites, only tooth, to be able to evaluate, construct the entire genome of ancient hominins, that the predecessor to Homo sapiens of 45,000 years ago exists. So I do not know what his perspective is. Apparently, from my reading, the deep cavities of tooth are preserved because of the buildup of calcium and etc. that is natural in the human body, and therefore they can be extracted. David Reich's lab does this every day, and there are hundreds of people who have done this. So I do not know why he said that, but something to look into. And I think there are experts that you could invite to have a look at this. But the conversation 2019 is there are 4 well-preserved ones that could be analyzed.
Thank you. I think Daisy.
Hello.
Daisy.
Hey, thanks, Bama. Thanks, Dr. Bama. The nice presentation, sir. I really appreciate it. My question is, the excavation of Rakhidigiri is belong to which halogroup? Why halogroup and mitochondrial halogroup both? I like to know, as well as the Indian Red Indians of Native Indians of America, which halogroup they belong? Thank you, sir.
Okay, the first question, it was a female. So females do not have Y haplogroup. They don't have Y chromosome, but they do have mitochondrial DNA, and that was not in their publication. I haven't polled Professor Reich to find out whether they did that, but they might have been focused totally on the genome part, because that was the most important part for trying to figure out their heritage, right? So total. So that is one question. The Red Indians have haplogroup Q, if I remember right. Chinese O, if I remember right, but there are O in India too, because there's back migration from Burma into Assam and then into West Bengal. I hope that answers your question.
Thank you, sir.
Hello. Hello, can I speak?
Yes.
Yeah.
Hello.
Yes,
Okay,
go
right.
Right, thank you. So I'm Dr. Kannan, a former colleague of Mr. Arasu. My question is, you just briefly mentioned about languages. Do you think that, my question is whether the Sanskrit and Tamil, where did they come from? Like the Tamil origins. Is it from the Middle East, or is it in the South?
Now you asked a linguistic question. That's another call.
I'm sorry about that. Yeah.
I'll briefly remark the following. There are linguistic features that connects Tamil Nadu all the way to Rajasthani. And there are linguistic features that connect Tamil to Elamite and to some aspect Sumerian language. So now the number of people working on the Elamite, only about a year ago, the linear Elamite script was deciphered by Professor Desert from Paris only about a year ago. And there are only about 10 or 11 inscriptions available. The Elamites were subservient to the Mesopotamian rulers, Sumerians. And so they wrote in their script, which is the cuneiform, and there are some phonetic features that is absent. And so that area is yet to be dug up, and not many people are working in that space, but it's a very important area. Professor Southward at U of Pennsylvania has published a work, and if you talk to him, he'll tell you the linguistic features of Marathi, Gujarati has Dravidian foundation. And the example he'll cite is the difference between inclusive V and exclusive V, nam, nangal. That is, nam means including you, nangal means we don't include you. That linguistic feature is present all the way into Rajasthan, but it's absent in Sanskrit and Hindi. It's present in Gujarati and Marathi, Old Canada. It's present in Tulu, Malayalam, and of course in Tamil extensively. But now if you go to Andaman, they have a more complex structure. They have a plural, singular, inclusive, and exclusively more sophisticated linguistic features. So in one way of saying, this is now a speculation, the Tamil we speak is a highly simplified version of the very ancient Tamil that was spoken 70,000 years ago, possibly. So
yeah, thank you.
That aspect has to be explored. It is still an open question.
Yeah, and he also beautifully mentioned about the language and the genetic group. And so the question in Tamil Nadu mostly is, யார் தமிழன் யார் ஆரியன்? இந்த ரெண்டு கேள்விதான் ரொம்ப முக்கியமா திரும்ப இந்த பேச்சில் வருது. அப்ப உங்களுடைய ஜீன்ஸை பார்த்ததுல இருந்தும் பிளஸ் டாக்டர் தங்கராஜுடைய ஸ்பீச்சை கேட்டதுல இருந்தும் what I derived is that the Indians are kind of a rainbow people with a total mixture of various genetic groups. And so what do you think about that? So
we are a blend, and according to Reich's work, because you can do mathematical model of the current day genetics and knowing the genetics of people that lived in older times and deconvolute. It's a technical term, and people who are in chemistry would recognize this. Bama will understand when you have a spectrum that composed of the composition of combined of two spectra, you can deconvolute using mathematical techniques, and that was developed around 60-70 years ago, and that has been done. And he comes to the conclusion that the onset of endogamy, that is marrying only within own cultural groups, occurred around 300 AD, approximately 1700 years ago. Before that, they just married whoever they met or whatever the case is. And so the separation of communities seem to have a late onset in the Indian peninsula. He compares that particular genetic phenomena with what happened in Europe, and there it did not take place. If you were to take 100 Europeans, they would have a genetic profile which is a blend. You cannot segregate them. But if you take, as I showed in a couple of tables, if you take a population of 100 in Tamil Nadu or anywhere in India, you'll be able to separate them genetically, if you will, based on their heritage. That is less likely in an open society compared to this. The other remark I wanted to say is I wonder, this is a speculation, not data. Manusmriti is written in pristine Sanskrit. So it's ascribed to Gupta period, approximately 300 BCE, 400 BCE. That origination of that Shastra, one of the 16 major Shastras, seem to coincide with genetic argument. Whether there is a correlation or not is for the scholars to determine. I'm just making that observation.
Oh, okay. Interesting. Thank you.
One
Dr.
question, sir.
Muttarasan, one piece of information. This is regarding there's a lot of studies has been done about what is generally called as a Dravidian marriage system or kinship terminology or kinship system. This is supposed considered to be internationally one of the unique type of marriage of getting married into the this kind of marriage within the families and all. And then, as you rightly pointed out, at some point of time, in the North Indian system, they are aghast to think about that Thai Maman in Tamil Nadu, even Brahmins marry the Thai Maman and all. But in North India, they go for Kanyadan. So always take the female from the not related. And so is there anything will have an impact? Because I know that we are talking in a very thing, whether this marriage system, after it comes established, will have any kind of say in this, the way the genetics moves?
Do not know. I don't have a good answer for that, because that is a cultural feature. For us to get a good answer for it, you need to be able to do what I would call a vertical genetic study. That is, you need to look at we need to look at a group and all their descendants, and then look at how they are related, etc. So I don't have a good answer for that question. A good question.
Okay, thank you.
Hello, sir. My question is, I'm Subramanian. Hello, sir. My question is, since it is a scientific treatment to your research, you have shown some sculpture images to connect the human races. It is an artistic expression only. How can you connect with the scientific conclusion? That is my question. It may be layman question. Thank you.
Could you say that again in a different way, because I don't think I understood your question.
Yeah, well, while your lecture, so you shown some images, some sculptures to connect the Iranian faces and the niggers faces, you know.
Okay. Now
that sculptures, yeah, it's all artistic expressions only. Yeah, how can you connect with your scientific conclusion for the artistic expression?
So everything that I presented in terms of data and interpretation have been the results of scholarly work, and where I have tried to interpret, I try to say that's my speculation, because I'm a trained scientist, so I'm careful about how I use my words. So with respect to the connectivity of cultural features to genetics is an entirely different topic altogether. For example, the marriage system that Balakrishnan mentioned is really a cultural feature, not a genetic feature. But if you want to see whether that feature exists, you really have to do a very comprehensive study of a particular population group, I think, for you to even attempt to answer that. No good answer for that question. Sorry about that.
One question, sir. You know, on the face of Earth, where can we find pure Aryans?
So that question begs another question. Who is an Aryan?
Yes, that's another one.
Okay. Now, remember, the term Aryan has been used to describe associated with people. For example, the haplogroup R breaks into R1 and R2, and then R1 itself becomes R1A and R1B. And R1A is the folks that migrated through steps into India. On the other hand, R1b migrated into Northern Europe. So my question, in a sort of a rhetorical question, is R1a Aryan or R1b Aryan? Because the evolution continues to take place, right? So that you can say a person has an Aryan descent, meaning people who belong to R1 branch. By the way, R2 appears to be totally native to India. They don't seem to exist elsewhere. And they're not directly related. They were earlier haplogroups. There are some theories about them that's not well explored area as yet. They are R2 haplogroup, but R1 is very well explored because substantial part of ancient DNA genome that's available to us is European and Russian. And most of the people belong to R1b in Europe, good number of them. And so a substantial research effort has gone in that direction, and it's very well characterized. That's not the case with R2, not the case with many other haplogroups, simply because we don't have data. We don't have ancient DNA data. India could play a major part in this because India has pretty much representative from all major haplogroups in different parts of India. And so the opportunity is there, but that hasn't been taken up for whatever reason.
Thank you, sir. Thank you.
Some of you had asked how can we contact Professor Raj through email or some other forums. We have a WhatsApp group for this JOC study circle. You might subscribe to this WhatsApp groups if you are interested in this kind of discussions. You can put down your number here. We can add you to the group. Maybe click. We are almost 2 hours into this lecture. As a concluding remark, Dr. Arasu and Balaya, we want to add up something more.
Hello. Once again, I thank you, Raj. It's an excellent talk. This is available in YouTube, so we can watch again and again. Bama's talk visiting Lothal and Dolavira also is available. And if you can suggest any topic like this surrounding the Indus Valley civilization and the Vaigai, the whole thing started from Dr. Balakrishnan IA's book, Journey of a Civilization, Indus to Vaigai. And we are following the conversation. And Roja Muthaya Research Library, which published the book, and Dr. Sundar Ganeshan is also here. So we will continue, and please watch the talks and spread this to your friends. Let them also talk. Here it is based on science, that's all. Science, scientific data. That's the basis of the whole thing. Thank you.
Yeah.
Wow.
Balayan, yes.
Vanakkam, Nandri, Sodratha, thanks a lot. It's been a phenomenal. Thank you very much for this illuminating talk. I think as Selaya sir mentioned that we should continue this conversation. Ultimately, our journey is for finding the truth, and we need not have any preconceived notions. That's how Monica also is there. Thank you for helping. So thanks a lot. It's been a phenomenal talk. At least I never heard on this subject such an illuminating talk so far.
Yeah.
Thank you very much. We should explore linguistic connectivity. Yeah. And I think there is some rich data there, I think. But anyway, exactly. That's something to be considered for future.
Yeah, I have a question. Like last time during my talk, we ended that lecture saying "Saaptingla" அப்படினு கேக்குறதுலயே ஒரு genetic heritage இருக்குன்னு சொல்லி முடிச்சீங்க. Can you please add more on that?
Well, this is my working hypothesis. I'm speaking like a scientist here, which is spoken linguistics that's embedded in the way we speak. I think the way we speak embeds in it cultural elements. And I'm not sure whether it's been explored. I haven't dug into it yet, but I've only looked at the verb syntax and certain features that are very unique to Southern languages, the combining of verbs together without and with a conjunction, which is very unique. By the way, Elamite also is written in that form. So is Sumerian language. And very fascinating part is, according to the Professor Desert in Paris, Elamite was the first phonetically transcribed language, the linear Elamite. And now every language in India is written phonetically, not English, not Spanish, not French. They rely on alphabets. We rely on phonemes. And that characteristics was from an inscription from, I think, 2000 BC, 2200 BCE from Elamite that Dr. Desert inscribed. I posted in our group. And so that is a major, I think that is a major connectivity to Elamite, to Tamil and all Indian languages, if you will. Sumerians didn't write phonetically. They wrote it using pictograph became symbols, and that was used to communicate. But Elamites wrote it phonetically.
Thank you.
சுந்தரையா
நீங்க எதுவும் சொல்ல விரும்புறீங்களா?
Sorry?
நீங்க எதுவும் சொல்ல விரும்புறீங்களா?
They put you on the spot, Sundar.
சார் ரொம்ப அருமையான லெக்சர் சார். எஸ்பெஷலி தியரட்டிக்கல் பேக்ரவுண்ட்லாம் கொடுத்துட்டு உங்களோட பர்சனல் டேட்டாவை கொடுத்து அதை நீங்க கனெக்ட் பண்ண அது வந்து ரொம்ப பிரமாதமா இருந்தது. அது ஆக்சுவலாக அந்த பர்சனல் டேட்டா வாஸ் வெரி வெரி இன்ட்ரெஸ்டிங். ஸோ ஐ திங்க் ஆல் ஆஃப் அஸ் ஷுட் ட்ரை தட்.
By the way, I just want to mention, என்னுடைய சம்பந்தி இங்க இருக்கிறாரு, ரோஷ். அவர் கன்னியாகுமரியில இருந்து வந்தவர். He happens to also belong to the same haplogroup. So he and I have a same common great grandfather. How his daughter and my son met is a different mystery, but there is a genetic connection with his family.
Thank you so much.
Thank you all. Let's formally conclude this session. It has been a very thought-provoking to us lecture. So thank you, Professor Raj Mutharasan, once again for this enlightening and eye-opening talk. Actually, as you rightly mentioned, there is more to go. This is a starting point. This is a tip of an iceberg is what we have just touched upon. And there is more to go. So let's continue this discussions in this forum and across all the forums. And thank you all participants. We had about 45 people attending this lecture. I saw that as the maximum number. Now we have 37. That shows the enthusiasm among us to know more and more and move forward along this scientific journey. So thank you very much for being a very active participant in this forum. Thank you all. Once again,
Madam, vanakkam.
Yeah.
Vanakkam, madam.
வணக்கம் எல்லாருக்கும் ரொம்ப மகிழ்ச்சி. என் கணவர் வந்து இவ்வளவு தூரம் படிச்சிருக்கிறத பத்தி எனக்கு ரொம்ப பெருமை. நம்ம தமிழர்கள் எல்லாத்துக்கும் பெருமை. அதான் எனக்கு முக்கியமான செய்தி.
I'm glad to share my PowerPoint slides for anybody who wishes it. You can send an email to me, or you can contact Arasu or Bama. We'll post it so it'll be available to you.
டாக்டர் சுந்தர் கணேசன் அவருக்கு ரொம்ப நன்றி சொல்லணும். இதுல உதவியா இருந்தாரு நம்ம டெக்னிக்கல் ஆஸ்பெக்ட்ல. நன்றி. RMR ரோஜா ரிசர்ச் லைப்ரரியிலிருந்து நண்பர் வந்திருந்தாரு. நன்றி. சுபு நன்றி. நன்றி சார்.
நன்றி, வணக்கம்.
பார்க்கலாங்க.
வணக்கம்.
Thank you, sir.
அந்த இதை எனக்கு அனுப்ப முடியுமா? ஏர் டிராப் பண்ண முடியுமா?
இதுங்க
பவர் பாயிண்ட் பிரசன்டேஷனை ஏர் டிராப் பண்ண முடியுமா?
ஏர் டிராப்
ஈஸி.
இதுல போடுறீங்களா நீங்க? நம்ம இதுலயே வாட்ஸ்அப் குரூப்லயே போடலாமா?
அதுல போட்டுறேன்.
பெருசா இருக்குமா?